So far, we've looked at examples of estimating a mean or a proportion for an entire group. What if we're interested in the difference between two groups? For example, we might want to know whether men favor a political candidate more than women, or whether customer service representatives have higher ratings after a courtesy training than before, or whether people from different racial and ethnic backgrounds have different blood glucose levels on average. In this session, we're going to look at three types of comparisons. Comparisons between two groups using the same measurement, comparisons in a single group using two different measurements, and comparisons between several different groups simultaneously. First, let's look at two groups using the same measurement. Some plant diseases spread quickly when the soil is particularly wet. Researchers were interested in conducting an experiment on bell peppers using two fields, but they wanted to know whether the soil in the two fields had comparable moisture content. The researchers took 14 moisture readings from the north field and 16 readings from the south field and calculated the percentage water content by volume for each. Looking at the measurements from each field, the soil in the north field appears to have slightly higher water content, but the researchers didn't measure every square inch of the two fields. If they had measured in slightly different locations, they would have gotten slightly different results. The researchers want to estimate how plausible it is that the north field has higher moisture content than the south field on average, given the selected measurements that they made. Moreover, they want to get a plausible estimate for how big the difference between the moisture contents of the two fields is. To accomplish this, they set up a Bayesian model for the water content in both fields. Recall that a Bayesian model has two pieces: the likelihood. This is the hypothetical probability distribution of the measurement values you would observe if a given model is correct, so to speak. And the prior. This is the plausibility of each possible model before you observe any data. When we were estimating the mean of a single group, we used a normal distribution for the likelihood and a vague uniform distribution as the prior for the mean and the standard deviation of that normal distribution. For estimating the mean of two groups, we can follow a similar pattern. We'll still use a normal distribution for the likelihood, but we'll need two different normal distributions, one for the first group and another for the second. Each of these normal distributions has its own mean, the mean for the first group and the mean for the second group, and each of these means will need its own prior distribution. Since the measurements are roughly on the same scale, we don't have measurements in the hundreds for one group and in the millions for the other, we can use the same range for a uniform prior for each mean. What about the standard deviation? We have two options. If we think that the two groups could have different levels of variability, we could use separate standard deviations for each group. On the other hand, if we believe that both groups are likely to have similar levels of variability, we could use the same standard deviation for both groups. Using the same standard deviation creates a simpler model, but it involves an assumption that may not be quite right. If your sample size is large, it's often best to use separate standard deviations for each group in order to avoid making the extra assumption that they have the same amount of variability. However, it's difficult to estimate variability when the sample sizes are small. So if you have a relatively small sample size, say less than 30 observations in each group, your results may be more stable if you use the same standard deviation for both groups, as long as it isn't entirely unreasonable that they have similar variability. In our soil moisture example, since the two fields are in a similar part of the country with similar weather and of a roughly similar size and with roughly similar drainage patterns, it doesn't seem too unreasonable to think that the variability in moisture content across each field would be the same. And since we have fairly small sample sizes, only 14 samples from the north field and 16 from the south field, let's use the same standard deviation for both groups. Here are the estimated posterior distributions for the mean water content in the two fields, based on the Markov chain Monte Carlo simulations, along with 95% highest density intervals. Based on these results, the most plausible values for the mean moisture content in the north field are higher than the most plausible values for the mean moisture content in the south field. But the two highest density intervals do overlap. In other words, there are some plausible values for the average water content in the north field that are lower than some plausible values for the average water content in the south field. It's conceivable that despite these particular samples, the south field actually has a higher average water content than the north field. It's conceivable, but how plausible is this? To find out, we can count up the number of times that our MCMC simulations had a higher mean for the south field than the north field. When we do this, we get just under 3%. So it seems fairly implausible, based on our sample, that the south field has a higher average water content than the north field. Now that we've established that we're fairly confident that the north field has a higher water content, our next question should be, how much higher is it? Some differences may not be very important from a practical perspective. Even though our Bayesian model did not have an explicit parameter for the difference between the two groups, we can estimate the posterior distribution for the difference between the two groups from our MCMC samples of the two group means. We simply calculate the difference between the two group means for each of our MCMC simulations, and we treat these calculated differences as if they had come straight from the original MCMC chain. When we do this, we find a 95% highest density interval from 0.05 to 3.49. All of the most plausible values for the difference in the moisture content are greater than 0, suggesting that the two fields do have different moisture content. But overall, most of the plausible values are relatively small. How small is too small to be important? This is up to the researchers, depending on the sensitivity of their research to small differences in the moisture content. In the previous example, we had the same measurement, water content, for two different groups, the north field and the south field. What about a scenario in which we have two measurements for the same group? Constructing highways across the wilderness can disrupt the migration patterns of many species. Wildlife researchers wanted to understand how a newly constructed highway was affecting deer populations that migrated across the planned path of the highway for the winter. They analyzed deer counts in 10 wilderness districts before and after the highway was built. In this scenario, we have just one group, the 10 wilderness districts. But we have two measurements for each wilderness district. We have a measurement before the highway was built and one after the highway was built. Each of the measurements before the highway was built can be paired with a corresponding measurement in the same wilderness district after the highway was built. In a scenario like this, we could construct a Bayesian model similar to the moisture model in the previous example. But if we did, we would be throwing out information about our measurements, namely that the measurements come in pairs from the same set of wilderness districts. How could this information help us? Wilderness districts have different characteristics that affect how many deer are present in the district. Things like the amount of tree cover, forageable food, water access, and so forth. Many of these characteristics won't change once the highway is built, so wilderness districts with above average deer counts before the highway is built are also likely to have above average deer counts after the highway is built, even if the average deer population increases or decreases across all the wilderness districts. In other words, the before and after deer counts are correlated. This is often, if not almost always, the case for multiple measurements from the same group of subjects. We can use this correlation to our advantage to increase our confidence in our estimate of the change of the deer population. There are a couple of ways to set up a Bayesian model for paired observations. The simplest way is first to compute the difference between the two measurements for each pair, and then estimate the mean of the differences directly. We'll use a normal distribution for the likelihood, and vague uniform priors for the mean and the standard deviation. Since the deer population could increase or decrease after the highway is built, the average difference could be positive or negative, so our uniform prior for the mean should include both positive and negative values. Standard deviations, on the other hand, are always positive. It isn't possible to have negative variability, so our uniform prior for the standard deviation should only include positive values. Here's the posterior distribution for the mean change in the deer population across wilderness districts. The most possible values are all less than 0, suggesting that we can be fairly confident that the deer population has decreased after the highway was constructed. How important is this decrease? We'll have to ask an ecologist to find out. I mentioned earlier that using the knowledge that our measurements came in pairs would increase our confidence in our estimate of the difference between the two groups. Did it work? Here are posterior distributions and 95% HDIs for a Bayesian model that treats the measurement as pairs, and a Bayesian model that ignores the fact that the measurements are paired, treating them as if they had come from two completely separate groups. Notice that the interval for the paired model is narrower than the interval for the independent groups model. Similarly, the posterior distribution is more concentrated on the most plausible value. This suggests that the extra information captured by the paired model helps us to be a little more confident in our estimate than the model that ignored this information. In this example, the difference is slight, but with measurements that are more highly correlated and with larger sample sizes, the extra confidence can really add up. Now, Why limit ourselves to just 2 groups? What if we wanted to compare several different groups at the same time? Nutritionists were interested in the effectiveness of 3 different weight loss programs: a low-calorie regime, a low-fat regime, and a low-carb regime. They conducted an experiment with 20 participants, randomly assigning each participant to one of the weight loss programs or to a control condition without a special diet. At the end of 8 weeks, the researchers measured how much each participant had lost under the program. In this scenario, we have 4 different groups, 3 types of special diets, and a control condition. And our goal is to determine which weight loss program was most effective. That is, under which weight loss program did participants lose the most weight on average? What kind of Bayesian model should we use to do this? In the 2-group case, we used a normal distribution for the likelihood with 2 different means, one for each group, and we specified a separate uniform distribution as the prior for each of the 2 means. We could certainly extend this by using 4 different means, one for each group, with a separate uniform prior distribution for each mean. But, just like we were able to get improved performance by taking into consideration the fact that paired observations were paired in the previous section with the deer, we can also get improved performance by pooling information about these diet programs across the different groups. Are these 4 groups related in any way? They aren't related like the paired observations were related. We don't have the same people in each weight loss group, and the people in each group themselves aren't related to one another. Nevertheless, the 4 means we want to estimate are related by the fact that they are all weight loss means. It's not like we're estimating completely different things, like weight loss alongside change in height or IQ and income. Even if the 4 weight loss programs have different levels of effectiveness, it's likely that they all have generally similar values. It isn't that likely that one program has an average weight loss of a few pounds and another program has an average weight loss of dozens or hundreds of pounds. We can use the similarity of weight loss in different programs to share information, so to speak, between the different groups. That is, even though the weight loss under the low-fat diet doesn't tell us exactly the weight loss under the low-carb diet, the amount of weight loss under the low-fat diet still gives us a general idea for how much we might be able to expect under the other weight loss programs. How do we express this in a Bayesian model? Instead of using 4 separate prior distributions for each group's mean, we can have all 4 means come from a common distribution, such as a normal distribution. The normal prior distribution for the group means has an overall mean, this is a measure of central tendency for weight loss programs, and a standard deviation. This standard deviation reflects how much variability there is between the weight loss programs. If this standard deviation is small, all the weight loss programs get similar results. On the other hand, if this standard deviation is large, the results for different weight loss programs are rather different. What values do we use for the overall mean and variability between groups? Well, we don't really know offhand. We'll have to estimate those from the data, which means that they will need their own prior distributions in our model. So for our Bayesian model, we have a normal distribution for the likelihood, that is, given a particular weight loss program's average weight loss, we think the specific weight loss for an individual who follows that program will follow a normal distribution. Then we specify another normal distribution as the prior for each of the group means. We'll need priors for the overall mean and the variability in the group means, as well as a prior for the variability of individuals within each group. We can use vague uniform distributions for those priors. Here are the posterior distributions for the mean for each weight loss program. Overall, the low-calorie regime seems to have done the best, and the control condition with no particular diet had the least weight loss. But there's a fair amount of overlap in these distributions, particularly for the low-fat and low-carb diets. How much are these diets really differing? When analyzing differences between several groups, sometimes we want to know which group is highest or lowest. Other times, we want to know how much better or worse one group is than another. These differences between groups are called contrasts. A contrast is the difference between 2 group means in a multi-group analysis, or it could be the difference between 2 combinations of group means. For example, we could talk about the difference between the low-calorie diet and the low-carb diet. Or we could look at the difference between the low-carb diet and the control condition. Or we could look at the average between all 3 special diets and compare it with the control condition. Finding the posterior distribution for any contrast from a set of MCMC samples is fairly straightforward. you simply compute the difference specified by the contrast, such as the difference between the low-calorie diet and the control condition, for each of the MCMC samples, and then use the computed differences as an estimate of the posterior distribution for the contrast, just like you would with samples for the mean. Based on the differences computed from the MCMC samples, we can find and interpret an estimated HDI for each contrast. Here are the 95% HDIs for several possible contrasts in our weight loss example. Based on these contrasts, the average weight loss for the low-calorie diet is probably somewhere between 2 and 7 pounds greater than the average weight loss with no particular diet. The weight loss for the low-calorie diet is also several pounds higher than the average weight loss for the low-carb diet. The low-carb and low-fat diets, on the other hand, have much more similar average weight loss. The HDI for the contrast between the low-carb and low-fat diets includes 0, suggesting that even if these diets do have different weight loss, it's plausible that the difference isn't very big at all. In fact, the posterior probability that the low-carb diet has a higher average weight loss than the low-fat diet is really only about 60%. The HDI for the contrast between the low-fat diet and the control condition also includes 0, but it only includes a few negative values, and the HDI also includes many positive values. This suggests that even though it's somewhat plausible that the low-fat diet isn't any better than no diet, it's also plausible that the low-fat diet has a small advantage over not dieting. In fact, the posterior probability that the average weight loss under the low-fat diet is greater than the average weight loss under the controlled condition is about 90%. That means that it's really quite plausible that the low-fat diet has greater weight loss than the control condition. But having a high probability of being greater than 0 doesn't mean that the difference is all that great. The advantage of the low-fat diet over the control condition, while likely positive, is still much smaller than the advantage of the low-calorie diet.